The third macromolecule which you find in the living organism are proteins. Proteins are also macromolecules, biopolymers made up of monomeric units, and these monomeric units are called as amino acids. And there are different type of proteins you find in nature. All these proteins are made of 20 different amino acids. And these 20 different amino acids are linked in different orders, forming all the different type of proteins. Proteins play a major role in building the cells. They also do various functional roles like acting as hormones and enzymes, and they do both the structural and functional work in the cell. Based on the amino acid functional group, there are different type of amino acids. So if you take the basic structure of an amino acid, there is an alpha carbon atom. To one end, it is bound to an amino group and a carboxyl group. The other end to a hydrogen and a functional group. So this the basic structure is same in all the amino acids, but where the amino acids differ is based on the functional group. So based on the functional group, each amino acid gets its property, and they can be categorized as non-polar, polar, or electrically charged acidic and basic amino acids. All these properties they get based on their functional group. The fourth type of macromolecule which we find in the living organisms are nucleic acids. They are also big molecules, macromolecules. There are two types of nucleic acids. One is this DNA and RNA. If you take the major role of these nucleic acids, they store hereditary information. In these two molecules, DNA and RNA, DNA is the molecule which stores the genetic material, which acts as the genetic material. RNA is doing various other roles. It also acts as an enzyme, and it is part of many proteins. Few viruses have RNA as the genetic material. Nucleic acids, they are polymers, and what is their monomeric unit? Monomer is a nucleotide. What is this nucleotide? So here it is a big molecule. So big molecule, it is going to be linked of small monomeric units. This monomeric unit of DNA is called as the nucleotide. So what is this nucleotide? Is this, that is, it has a 5 carbon sugar. Okay, a 5 carbon sugar. The first carbon is linked to a nitrogenous base, and the fifth carbon, 1, 2, 3, 4 and fifth carbon is linked to a phosphate group. So all these three structures together make up a nucleotide. And you also come across a term called as nucleoside. So what is it? It is having only the sugar molecule and the nitrogenous base and no phosphate is called as this nucleoside. If you take the DNA and RNA, the nucleic acids, both are having this monomeric unit as the, what is the monomeric unit is this nucleotides. So where they differ, if you take in the case of DNA, the 5 carbon sugar, you can see it is a deoxyribose. It is having 5 carbons, but here deoxy, somewhere oxygen is missing, at which position is at the second carbon position. Usually it has a OH, but this O is missing, so this 5 carbon sugar becomes a deoxyribose sugar. So the nucleic acid which has this deoxyribose sugar is called as the deoxyribonucleic acid, DNA. If you see about the nitrogenous bases which are linked at the first carbon of the 5 carbon sugar, in DNA, if we see the nitrogenous bases found are adenine, guanine, thymine and cytosine. These nitrogenous bases are categorized as pyrimidines, and as purines. Pyrimidines, you can see the structure is similar to that of a benzene ring. They are heterocyclic aromatic compounds, and they have nitrogen atom at the first carbon and the third carbon position. Thymine and cytosine are pyrimidines. The purine nitrogenous bases, they have this pyrimidine ring fused with imidazole ring. So it is also a heterocyclic aromatic compound containing a pyrimidine ring fused with an imidazole ring. Adenine and guanine are purines. And in DNA, these are the nitrogenous bases found, that is adenine, guanine, thymine and cytosine. But whereas in the case of the other nucleic acid RNA, it has adenine, guanine, cytosine, but instead of thymine, there is another nitrogenous base, which is this uracil. If you see the DNA, we saw that both the DNA and RNA, they are made up of this monomeric unit nucleotides. And if you see the structure of the deoxyribonucleic acid, it is a double helical structure. That is, it is not just one strand of nucleotide, but it is containing two strands of nucleotide. And if you see these two strands of nucleotide, they are helically coiled, forming a double helical structure. We will see in detail about this structure. So to study some of the history behind this discovery of DNA, Walter Sutton discovered chromosomes, and he found that they are made up of, chromosomes are made of DNA and protein. And but however, the scientists were not sure which one which one is actually the genetic material, whether it is the DNA or the protein. Then Frederick Griffith in 1928 showed that DNA was the cell's genetic material. Rosalind Franklin took diffraction X-ray photographs of DNA crystals, which helped in studying the genetic, that is the DNA model. Watson and Crick in 1950, they built the first DNA model. So from all this research, we were able to find out the structure of the DNA. So DNA is a macromolecule, so made of small units, that is the monomeric unit, which is called as the nucleotide. And if you see these nucleotides, they are made of phosphate. It consists of phosphate group, ribose sugar and a nitrogenous base. And in DNA, if we see, the nitrogenous bases found are adenine, thymine, guanine and cytosine. And when we see the structure of the DNA, we find that there is a complementary base pairing. It is always a purine binds with a pyrimidine only. Not two pyrimidines does not bind here. Only a purine will always bind with pyrimidine. And we also see that A is always pairing with T and G pairs with C. This has been explained by a rule called as Chargaff's rule. That is Erwin Chargaff, a scientist, showed that the amount of the four nitrogenous bases A, T, G, C, that is, if you see the amount of adenine and thymine, it is of the same amount, and guanine and cytosine, they are of the same amount, which indicates that adenine pairs only with thymine and cytosine pairs only with guanine. How do they pair is forming hydrogen bonds. And the number of hydrogen bonds between adenine and thymine is 2 hydrogen bonds, and cytosine and guanine, we see 3 hydrogen bonds being formed. So we told that the DNA has a double helical structure. If you take one strand, what is this one strand made of? You can see here each nucleotide, one nucleotide is linked to another. So this is one nucleotide where it has a 5 carbon sugar, a nitrogenous base and the 5th carbon of phosphate. So this is one nucleotide. It is linked to the next nucleotide. So what is the linkage? Linkage is a phosphodiester bond. So here the fifth carbon you have a phosphate. This phosphate links to the third carbon of the other nucleotide, forming a phosphodiester bond. So what is the linkage between the monomeric units in nucleic acid is phosphodiester bond. Okay, we know how one strand is made, but how does these two strands, how they are linked together to form this double helical structure? So if you see here in this in the sugar phosphate backbone, the nitrogenous bases actually protrude from this sugar phosphate backbone, forming hydrogen bonds with the nitrogenous bases from the other base pair, other sugar phosphate backbone. So the two strands are linked together. This is strand 1 and this is strand 2. Let us take. These two strands are linked together by hydrogen bond between the nitrogenous bases. And if you see the structure of the DNA, we told there are two strands. One strand runs in one fashion and the other in the other fashion. That is why we tell that they are anti-parallel strands. That is, in one end, if you see, will be exposing a phosphate group. In one strand, let us take, one end will be exposing a phosphate group and the other end will be exposing this 3' end of the 5 carbon sugar. So we tell that the strand is 5' to 3' end. If you see the other strand, it will be in the opposite direction. This end will be having a phosphate group and the other end will be having a 3' group. So the strands are running in anti-parallel fashion. DNA has a double helical structure. The one DNA strand having polynucleotides linked together. So that polynucleotide strand is having a sugar phosphate backbone. And here, because of the complementary base pairing, the two strands are linked together by hydrogen bonds. And if you take the two strands, they are running in anti-parallel fashion. So they are anti-parallel strands running in opposite directions. We learnt about the structure of DNA, and when we compare with the other nucleic acid RNA, so DNA is a deoxyribonuclease. Why it gets this term? We saw that in the 5 carbon sugar, the second carbon there is no oxygen. So that is why no oxygen is found. So it is called as a deoxyribo sugar. If you take in the case of RNA, in the nucleotide, okay, it has a ribose sugar, a normal ribose sugar. At the second carbon position, it has a OH group. So the RNA has a normal ribose sugar, so it is called as ribonucleic acid. And what is the difference here from DNA is in the case of RNA? So it differs in the 5 carbon sugar where it has a normal ribose sugar, and the other thing is based on the nitrogenous bases. In DNA, it has adenine, thymine, guanine and cytosine. But whereas in the case of RNA, instead of thymine, it has another nitrogenous base, uracil. And if you see the structure of DNA and RNA, DNA is a double helical structure, but whereas RNA is a single stranded structure. And if we see the RNA, there are different types of RNA based on the function what they perform. So the first type is this mRNA. mRNA, it gets its name from the function, that is, it carries message, information for the protein. So it is called as a messenger RNA. And ribosomal RNA or rRNA, it is a part of ribosome, so that's why it's getting its name ribosomal RNA, rRNA. And transfer RNA is the third type of RNA. Its function is to transfer the amino acids to the site of protein synthesis. So it is transferring amino acids, molecules, so it is called as the transfer RNA or tRNA. We will see in detail about each of these RNAs. The first one is the messenger RNA. or in short form we call it as the mRNA. So this carries a message, so message for a specific protein. And if you see the length of this RNA, it is made up of 500 to 1000 nucleotides long. And how is the information present here in the mRNA is as a 3-letter code. And this 3-letter code is called as the codon. So it is a long strand of nucleotide, but how the information is read as 3-letter code. AUGS1. So this gives a meaning. What is the meaning is meaning for an amino acid. The next 3 letters, when it is read, it gives a meaning. That is meaning is for the amino acids. Transfer RNA or tRNA is made of 75 to 80 nucleotides. They have a specific structure called as clover leaf structure because they have a structure very similar to that of a clove. In the clover leaf structure, it has a site where the amino acid will be attached to, and it has an anticodon a site where this anticodon will recognize the appropriate codons on the mRNA and bond by with the help by forming hydrogen bonds. So this tRNA will carry amino acids. Once the tRNA which carries amino acids, they are called as charged tRNA. So they carry the amino acid and transport it to the mRNA where the protein synthesis is taking place. There are 4 ATP molecules required for each amino acid to be added in the polypeptide chain. 2 ATP molecules are required for tRNA to carry, that is to charge the tRNA. Charging is adding an amino acid to the tRNA. So for adding amino acid to the tRNA, two ATP molecules are required. One to carry the charged tRNA to the ribosome and one to move the ribosome to the next codon. The third type of RNA is the ribosomal RNA. So they are a part of this ribosome. So that's why they're getting the name ribosomal RNA. And if you see the length of this ribosomal RNA, usually they are 100 to 3000 nucleotides long. These are made inside the nucleus and they get associated with the proteins to form the ribosomes. And here if you take these ribosomes, they have a larger unit and a smaller unit. And if you see in the ribosome structure, 60% of it is made of proteins and the rest of 40% is this rRNA. And if you take a ribosome, it has 2 sites. One site is the P site and the A site. To summarize about the macromolecules, we have 4 major macromolecules in the living organisms. One is the carbohydrate, lipids, proteins and the nucleic acids. The functions of carbohydrates, they are energy molecules and they also act as structural molecules in few organisms. And here the monomeric unit of carbohydrates is sugar molecule. And few examples: glucose, fructose, lactose, sucrose and starch. And lipids are hydrophobic molecules, the next type of macromolecule. They are usually long-term energy storage molecules. And the monomeric unit, one type is this fatty acids. Examples of lipids are fats, oils, steroids and phospholipids. The third type of macromolecule, very important macromolecule, is this proteins. They have various roles. They act as enzymes, as structural compounds for storage, transport, and various functions are performed with the help of proteins. The monomeric unit of proteins are amino acids. Nucleic acids are the other type of macromolecules. The major role of nucleic acids is to store the genetic information. The monomeric unit of nucleic acids are nucleotides. The types of nucleic acids are DNA and RNA.